Go ahead and get started. Welcome everyone to the usual Thursday DMICE Informatics Research Conference, and we have this week, as we have many weeks throughout the year, a student presentation. We're going to hear from Jeffrey Hunter as he does his master's thesis proposal defense, which means that he's going to present his proposed master's thesis work to us, and we will give him comments and feedback, and then when the general audience is done, his committee will sort of stay a little afterwards and further work out the details with him, and then once he's approved on that process, as many of you know, we'll be able to go on and do his master's thesis. So the title of his talk is Expression Variations Associated with Influenza Infection Severity in Humans and Mice. As a human who's just getting over something that was probably influenza, I'll be interested to hear, although I don't think my genome's been sequenced yet. Go ahead.
Maybe.
Yeah,
there we go.
It's mostly done for the broadcast.
Yes. Thank you. So, like you said, my name's Jeffrey Hunter, and I'm talking about my proposal for my thesis, which is looking at the expression variation associated with influenza infection. So, to start with, the World Health Organization estimates that 5 to 10% of adults and 20 to 30% of children are infected with influenza annually. Of these, there are an estimated 3 to 5 million severe cases, which can result in between 250 to 500,000 deaths each year. These infections are, of course, caused by the influenza virus, of which there are 3 types, A, B, and C. The B and C viruses only affect humans, while the A viruses are able to infect a wide range of hosts. Additionally, there are several subtypes of the A viruses, which are categorized by 2 viral proteins, hemagglutinin and neuraminidase. Currently, of various subtypes, H1N1 and H3N2 are the most common in humans. For a general uncomplicated influenza infection, symptoms generally include fever, cough, sore throat, muscle pain, nasal congestion, things like that. In order to be classified as a severe infection, however, there's 4 criteria that the World Health Organization defines. The first is if a patient presents with any specific complications. These can include lower respiratory tract disease, such as shortness of breath or pneumonia, central nervous system involvement, such as encephalopathy or encephalitis, secondary complications including respiratory failure, septic shock, or multiple organ failure, and other complications such as dehydration or myocarditis. Additionally, if a patient has any sort of chronic condition that's exacerbated by an influenza infection, their infection would also be classified as severe. Likewise, if a patient has any other condition either caused by or in addition to an influenza infection that requires hospitalization, that infection is also severe. And lastly, if a patient presents with what is initially an uncomplicated infection and then progresses into something more severe through one of the other categories, it's considered that their entire infection was severe. So from these, it's pretty clear that there's significant variation in the symptoms observed, and unsurprisingly, there's also a large number of factors that can contribute to the range of symptoms and severities experienced. As one might imagine, both the strain and quantity of virus one is exposed to certainly plays a role, as do plenty of environmental factors around patients. Additionally, there are quite a few differences between patients that lead to these. Age is definitely strongly correlated, with the very young and the very old being much more likely to have severe infections. Chronic conditions obviously play a role in the rate of severe infections as well. And there also does seem to be quite a significant genetic component as well. So this complexity underlying an influenza infection has made it rather difficult to tease apart the contributions of the individual components that are basically going into this infection severity. So what we'd like is the use of a more controllable model that can potentially shed light on each of the components a bit more readily. One of these that's been commonly used in biology and influenza specifically has been inbred mouse lines. They've allowed for identification of several genetic factors so far, but unfortunately don't accurately represent the genetic diversity found in an outbred human population. In order to combat this, the recently developed collaborative cross makes use of 8 parental strains that are outbred in a controlled manner in order to more accurately represent human genetic diversity while still maintaining quite a bit of the reproducibility that was the hallmark of the inbred lines. In order to make use of the data from the collaborative cross, then, it's very important for us to have computational tools that allow us to access and understand the data that we've collected. Much of this is done in the framework of ontologies, which are structured in controlled vocabularies that basically describe and annotate a body of knowledge. They work by defining the vocabulary and the structure of the ontology, and then defining the relationship between each of those objects. For many of the ontologies out there, they're divided definitely by species and often by information as well. So, for instance, the human phenotype ontology and the mouse phenotype ontology are very separate and different entities. While the terms and relationships within each ontology are fairly consistent, there are much greater differences between the different ontologies. This has, unfortunately, limited the ability to integrate data across species and has only compounded difficulties caused by different conceptualizations of phenotypes and species-specific anatomies. There has, however, been some progress in the last few years in being able to map related terms in different ontologies. The Monarch Initiative, for instance, has established tools that allow for some computational comparison between species and involve mapping related terms in different ontologies and making use of overarching superclasses to basically bridge the connection between them. Such mappings will be essential for my project here in order to integrate both human and mouse influenza infection data. On the human side of things, then, I'm planning on looking at expression data taken from the gene expression omnibus. The expression data sets have been looked at and selected. The ones that I'm wanting to use so far have to include influenza severity annotation with severity definitions equivalent to the World Health Organization guidelines. I need them to have the raw microarray data available, and lastly, to have virus data rather than, or potentially in addition to, vaccine data. From there, I needed to decide whether I was wanting to look at the important genes or try and go more broadly and look at pathways. Initially in this project, I was rather excited to try and find some sort of causal difference for influenza severity, but from looking at basically the data in the arrays, it quickly became apparent that it's probably not the most practical. The microarrays for the human data sets just can't capture all of the expression variation, and there are quite a number of different arrays that have been used in the data sets as well, which has limited the direct comparisons that I can make on the gene level. Additionally, as one of the main outcomes for my project is going to be basically establishing a framework for prioritizing further QTL studies and ongoing projects, it seemed a more generalizable application would be to branch out to pathways as well. To that end, then, I'm proposing the following specific aims for my thesis. Excuse me. The first is to identify public human data sets with influenza infection severity annotation, curate, analyze, and integrate these data in order to identify key pathways, genes, and regions implicated in influenza severity and response to infection. The second is to establish a mapping of human influenza infection severity phenotypes and genes in the mouse model, in this case the collaborative cross, and ensure that these mappings can be extended to other mouse models. Lastly, I plan to use the established collaborative use the established cross-species phenotype and candidate gene mappings in order to provide a computational framework to guide prioritization in collaborative cross-studies for identification of genetic and gene pathway associations related to influenza infection severity. My proposed research then is focused on mining human phenotype and expression studies, and ultimately will provide guidance and a computational framework for identifying candidate genes in collaborative cross-mapping studies. Currently, I've curated an initial set of human data sets from GEO, and having done so, I now also have an analytical pipeline established so that if I come across more in the course of this project, I should be able to quickly add them to the existing data sets. To go into a little more detail on what I've been doing, well, this one is just sort of a general flow of the project. Basically, I'm looking at it sort of as two parts. The first is working with the OHSU ontology group to establish more of the ontology mappings and extend the Monarch initiative in order to relate the data between the human and mouse data. And the second, for the statistical analysis, I'll be using R and several packages from Bioconductor in order to identify differential expression and overrepresented pathways. For the ontology mapping part, I will need a few pieces of information. The first, obviously, would be the names of ontology terms that are related but don't yet have clean mappings. And in order to help make sure we identify the right ones and map properly, I'll also need to grab the parent terms for each. From there, I'll also need a textual definition to extend the annotations and a reference that I can cite between the terms. While it is possible for me to obtain and extend a local copy of the cross-species mappings, this presents two problems. The first is that it wouldn't particularly assist anyone else in future research along these lines. And the second is I'm quite liable to deviate from the controlled terms in those ontologies. Having spoken with a couple members of the ontology group, it sounds like they should be able to quickly add any mappings and help me extend the live copies pretty quickly. So if at all possible, my plan is to follow the best practices here and work with them to extend the live mappings. For the statistical side of things, it's again broken down into two parts. The first, for the human data sets, I've taken all of the published results from the data sets and recorded the significantly differentially expressed genes. Then, pulling in the annotations from the microarray platforms used, I've constructed a consistent table with gene name, directionality of the expression, as well as GenBank and NCBI IDs. That table's then gotten passed to R, and pulling in the GoStats package, I've been able to identify significantly overrepresented pathways, as well as the genes that were responsible for identifying those pathways. On the mouse side of things, the first step is going to be taking the more quantitative phenotype measurements that we have and establishing a concrete severe infection definition in order to stratify the uncomplicated versus severe infections. Once I have that established, I'll then pass the microarray data into R, and making use probably of the packages Lima and Affy, I'll identify the differentially expressed genes, as well as getting the IDs that I need for each. And then, similar to the human side of things, pull in GoStats in order to identify the overrepresented pathways and genes from the mouse data sets. With this project, the main limitation that I touched on a little bit definitely seems to be the microarrays. As I said, they're unable to fully capture all of the expression data, and especially on the human data sets, each one I've looked at so far makes use of either a different version or a different platform in each study, which definitely limits the direct gene-level comparisons I can make. To further complicate just the human data sets, the samples have unfortunately all been taken from different time points in each study. One of them was from a single time point, one of them has two, one of them was over the course of 4 to 5 days. So trying to get all of that to line up nicely may be a bit tricky as well. The other issue between the human and the mouse data sets is that the human studies have all made use of blood samples, while the mouse data has been collected with lung samples. There's unfortunately not much way to go back and either get lung samples from the humans or blood samples from the mice at this point, which is unfortunate, but hopefully we'll be able to make that come up with still useful and informative information. The other main limitation that I may run into would be issues with the ontology mappings being somewhat limited. Since extending the ontologies and the mappings may require some development with the groups maintaining the ontologies, it's possible it may take a bit more time than I have, given the short timeline of this thesis, but I'm hopeful I can get at least most of it lined up. So, at the completion of this project, it's my expectation that I'll have identified genetic and expression variation that's correlated, excuse me, it's correlated with differences in observed symptoms in mice and humans, and that I'll have utilized these to establish a framework for prioritizing findings of genetic and gene pathway studies related to influenza infection severity. In order to do so, I have the following timeline outlined. The first part has been to look at the human data sets, and that pretty much running out of time. I was allocating till the end of January to finish adding data sets to that. I'm currently working with the ontology group in order to extend the mappings, and after which I'll obtain the mouse data and begin to analyze those. At the same time, I've been performing literature review. We'll be working on the actual thesis document itself, and finally, the defense of the thesis. I fully understand that there are quite possibly going to be some delays in here, so I have also allocated a month between my intended conclusion of the project and the final deadline. And with that, I'd like to thank my committee members for their ongoing help, and thank you guys for your attention.
First of all, Jeffrey, thank you. It sounded like a really interesting topic, and you spoke very clearly. It was great.
Thank you.
I don't understand one thing, though. When you're looking at your human subjects' genomic data, what are the inclusion criteria for the study, and how are you going to avoid just getting indicators for severe chronic disease rather than what makes it differentially susceptible to flu? Okay, so you get severe influenza is, of course, much more likely in somebody who has COPD or heart failure or liver disease or something like that, for which there is a large set of genomic markers that we're working on already. Mm-hmm. How are you going to differentiate the things that make you more likely for severe influenza beyond that, or are you just going to come up with the markers for these diseases?
As of yet, I'm not entirely sure. Part of it has to do with the actual people who were taken. Most of the subjects for the human data sets have, basically they're selected based more on respiratory issues rather than other complications like that, which should help somewhat with that. So focusing more on like the respiratory failure part of severe infections rather than exacerbation of an existing chronic condition. Beyond that, it's a bit hard to say and definitely something I should look into. Thank you.
That's a great point. The problem is, and this is a real issue because it's bias in the literature, we won't know. Because when they describe the cohort, they won't necessarily tell us in the paper unless we contact the authors if they actually took that into consideration. Because all of the papers that we selected were based on the fact that they were studying severe influenza, and that was really the criteria that was described. So it's a really important point, but not just for his work. I think it's an important point in general because when we read these papers, right, that's what subset, had that component, and how good clinical education that we even have access to in secondary knowledge.
So could you talk a little bit more, or provide, an illustrative example of how you're going to use this ontology mapping to create the prioritization framework? Because we didn't really talk much about prioritization at all. So how can you, really understand what you're really going to be doing?
Yeah, that's one that at this point I haven't dove too much into the actual process that I'm going to be using for it. So I, at this point, don't have these are the steps that I'm going to be doing in order to create that. So the ontology mapping part is basically to relate that this phenotype, these genes, etc., found in mice can be mapped to these similar things in humans. But in terms of creating the prioritization framework itself, that's one that I'm still working on developing.
Talking about mapping the genes from humans to mice, or the symptoms or indicators of severity from one, or both?
Ideally both.
Because it seems like there's other methods of mapping the genes in terms of evolutionary divergence and things like that that would be more appropriate for, and pathways, overlap that would be more appropriate for the gene. At least when you started talking, I was thinking that the ontology mapping would be mostly for the phenotypic information.
And that is definitely what I'm more focusing on, but it would be nice to be able to have that for the genes as well.
So, I think the idea is that you can have classify your candidate genes based on the phenotypes in one species, and then use the mappings implicit in the ontology to help prioritize the candidates based on both phenotypic similarity and orthology. And then you could extend that to other pathway members depending on what kinds of relationships you might want to utilize for that prioritization. So that if you start out with 300 candidates that might be particularly relevant in human, that you could then, using the phenotypic similarity to the mouse, potentially reduce that number of genes of interest based on a variety of factors, including pathway membership or phenotypic similarity.
So there's always a danger in a bioinformatics student undertaking a study as clinical types can actually understand. And so my no, it's very interesting, and I totally understand what you want to do. And Dion actually asked the question that I was also going to ask, and Shannon emphasizes its importance. But another aspect of this, and I don't know how familiar you are with kind of the sort of biology around influenza, but what kinds of lessons would a sort of biological researcher who understands influenza and the genetics behind it and so forth be able to kind of take away from your study to pursue new avenues of research that would ultimately result in clinical observations?
Well, the main goal for it would be not so much to say that this is what's causing it, or I think what's causing it. It would be nice if I could come up with that, but I more expect to have something that basically allows you to say, you've done this study, come up with this list of possible genes that you think are interested. I have found from this data mining that this set of genes looks to be implicated. The ones that overlap are possibly more likely to be useful. Possibly consider focusing your research in that area. Yeah?
Can I try and respond to Dr. Hirsch's question?
Yeah.
One thing they're looking at doing is using the results of this type of study to identify what needs to go in the influenza vaccines. So we take and we identify what the genetic variants are for the people who are likely to have severe illnesses, and make sure we package the vaccine, so it's going to help those people be more resistant to flu.
I would just add to that, besides the precision medicine piece of this, which is a big issue, especially for what we consider to be vulnerable populations, where we've had groups that are really susceptible. That's one of the major concerns of infectious disease now is vulnerable populations. But the other piece of this, going back to Dr. Hershey's question or comment, is the prioritization piece, and also where the holes are. One of the important things that we will do, and I think that the Monarch Initiative has done, is tell us where are we missing things. What phenotypes do we need to be collecting? So for the basic scientist, wow, we can do these mappings, but here are potentially other phenotypes we could be collecting that we're not relative to some of these severe. And I think that's one of the things that has come out just from this initial perusal is where we have gaps, where we were not even collecting data that would allow us to map these phenotypes. And I think that's another important piece of that, besides the emphasis you've placed on prioritization.
Well, if not, thank you all for coming, and we can